tell us a little bit about your early interest in that field, and it started with a very hard science discovery of blue light. Were you interested in sleep before that, or what led to that discovery?
Well, I was interested actually in circadian rhythm in general because my view of biology at that time was the biology of time, how biological systems track time, remember time, and use it was kind of unexplored. So I started my career in plant circadian rhythms, and while studying plants, it became very clear that plants use different light sensors to sample whether it's morning, noon, afternoon, evening, or whether they're under the shade of another plant. So that was kind of fascinating.
How do plants do that? They don't have eyes.
In plants, what I understood or what we recognized was if a plant has any light-sensitive protein, the clock recruits that light-sensitive protein to entrain itself. And there was a cool insight, and that's the time when human genome was being done. People are finding new kind of proteins, and that's when melanopsin, this opsin or light-sensitive protein-like molecule, was also found in human genome. So then immediately we thought, well, we have seen that example that plant uses every light-sensitive protein to adapt to light-dark cycle, so maybe human and mouse will use this melanopsin to adapt to light.
And then in animals, are those opsins in the eye, or are they in other parts of the body?
Yeah, so the history of melanopsin, why it is called melanopsin also is very interesting, because it was originally discovered in the melanosomes, or the pigment cells in frogs. So if you put a frog in bright daylight, then frogs will kind of put their natural sunscreen, they'll become dark. And that's because of melanopsin sensing light and then signaling through melatonin. Again, the name melatonin comes from the hormone in frog skin or melanosome that changes its skin tone. So we knew that melanopsin in frog skin somehow may be linked to melatonin. So that was kind of the clue that maybe melanopsin might also entrain or reset melatonin either through circadian clock or indirectly. So that was kind of the background. And then around that time, since the genome was coming out, we knew that there are a lot of people who are competing. So I remember one day my PhD advisor, Steve Kay, came to the lab and said, who wants to make a melanopsin knockout mouse? And the competition is too tough, so the stakes are pretty high. And also our lab was not a mouse lab. So not many people took that bet, and I took the bet. I said, okay, I'll make the melanopsin knockout mice, and there was also my switch from plants to animals. And in the next year, simultaneously 3 different labs made this melanopsin knockout mice, and we all came out around the same time, connecting melanopsin to circadian entrainment. And that's why in 2001, when the stories came out, Science magazine kind of listed melanopsin as one of the top 10 breakthroughs of the year.
So today in the presentation, you hinted on some other ways in which that rhythm gets entrained, not just light.
Yeah. So in mice that we mostly work on, mice are nocturnal. So that means they eat at nighttime, prefer to eat at nighttime, and daytime they sleep. And up to say maybe early 2000, the idea was the brain clock regulates everything, and the brain clock is tied to the outside world through light. Then the question became, well, when we eat at the wrong time, then there are conflicting signal that the brain clock is trying to tell liver when to turn on and off, and then the food is giving a different signal. So we did a very simple experiment. We took mice, gave them food during daytime or nighttime, and we asked, what happens to the liver clock? Will the liver clock take instruction from the brain clock that's tied to the light-dark cycle, or the liver clock will directly listen to the food cue? And to our surprise, almost every clock-regulated gene in liver, including the circadian clock, it just followed food, not light.
So food was more powerful than light?
Yeah, so food was more powerful than light for all these peripheral clocks. Not the brain clock, the SCN, the master clock center in the hypothalamus, it still tracked light-dark cycle. But all the peripheral organs, even half of the brain outside the hypothalamus, they track food. So then it became very clear that if we have to synchronize and sustain robust circadian rhythm, then maybe we should also think about food.
Did those mice that had then opposite influences on their circadian rhythm, did they have poorer quality sleep? Was there a way to measure that?
Yeah, so that's actually nowadays that's used as a method to disrupt sleep during daytime. Now it is very well accepted that if you feed mice during daytime versus nighttime, even if the mice may eat the same number of calories, the day-fed mice will be less healthy than the night-fed mice.
So one thing that really struck me during your presentation at BrainMind that I hadn't seen before was the long-term effects on all kinds of very serious illnesses. Even, I know in particular you looked at Alzheimer's disease. What is the latest state of quality peer-reviewed research showing a link between lack of sleep over a lifetime and Alzheimer's disease?
So there are, of course, these are correlative studies that people who actually in mouse models first, by disrupting sleep, you increase the you advance the onset of Alzheimer's disease and also increase the severity. What will be interesting is to see whether time-restricted eating or any kind of circadian intervention can slow down or better manage those diseases. For example, even among Alzheimer's patients, we know that these patients are almost indoor. They have very little chance to go outdoor. So light, the lack of light becomes a big problem. So then the question is, how do we deliver light in old age homes? And if we deliver sufficient light from a horizontal angle, because the vertical illumination is not useful in this case, then what are the parameters we are looking at? Means we are not talking about curing Alzheimer's with light, but how do we improve the quality of life? How do we improve sleep at night? Because when these patients don't sleep at night, then it becomes a disease for two people because the caregiver also suffers. So I think those objective measure of endpoints will help to clearly define what is the impact of circadian optimization. And second thing that we are finding, surprisingly, is we never thought that time-restricted eating will have an impact on Huntington disease or Alzheimer's disease. And recently there are quite a few papers coming out showing that time-restricted eating delays the onset of Huntington disease or improves sleep. So that will be equivalent to improving the quality of life, but that's all in rodent model. And then another thing that I also mentioned is drug timing, because if circadian clock directly or indirectly regulates the targets of most of the existing drugs, then by matching the timing of the target and the drug, we can improve efficacy or reduce adverse side effect. And that field has pretty good history in cancer. So there are certain drugs for breast cancer that work very well in morning versus evening. So extending that to other classes of drugs is something that we hope to see. Right now we see that many pain medication, particularly arthritis medications, they can be optimized for time release because the pain is much more severe in the morning, but the medications, if they're taken in the previous night, then it better manages pain. Then the slow release formulation of these pain medications, when given at bedtime, they're released around 2 or 3 o'clock in the morning, so those work the best.